This module covers the basic concepts of unsupervised learning. In particular, we focus on a common and flexible non-parametric technique, that is, the k-means algorithm. In the last module, we dealt with supervised learning, that is, when, for each patient, we observe a feature, X, labels Y, and we aim to predict the label of a future patient that will enter the study. We now instead focus on unsupervised learning, whose setting is similar, but there are no true observable labels Y. As a consequence, our goal will also be different. This time, we aim to detect the underlying and unobservable relationships or patterns between the data. That is why we say that unsupervised learning is a descriptive tool, not a predictive one. Indeed, we do not aim to predict the output of a future observation, and therefore we do not need a training dataset. Let me repeat myself, we only search for relationships within a given dataset in which no new observation will be inserted. In this framework, the machine learning algorithm we will see today are model-free or non-parametric, meaning that there is not an explicit model as in regression or neural networks. The most common task in unsupervised learning is clustering. We say that a cluster analysis is the attempt of partitioning a set of objects in distinct subgroups named clusters. This means that each object, or point, or observation must belong to one and only one cluster. A good clustering is achieved when objects within each group represent homogeneous subpopulations, which means that they are similar to one another, while objects in different clusters are dissimilar. Such differences between different groups represent heterogeneity between such subpopulations. In the picture we see an image segmentation where each color represents a different cluster is displayed. But what does similar mean? In general, we say that objects are similar if they are close according to some notion of distance. The simplest case is when we use the Euclidean distance, hence objects that are closely located in the space. But generalizations are possible. For example, in the previous slides, pixels are not clustering according to their location on the brain, but according to how similar they respond to urges. Hence, the most suitable notion of similarity is usually chosen by experts according to the problem to be solved. Cluster analysis is particularly useful in medicine because it allows effective and efficient treatment by personalizing the therapy to better suit patients' profiles. Indeed, it is common to observe heterogeneity in patients' responses to treatment. Cluster analysis models such heterogeneity and finds subgroups of patients which are instead used to respond in an homogeneous way. K-means is the first algorithm we introduce to perform clustering. Let us introduce it through an example, hence consider the scatter plot on the right of the slide. It represents quantities measured in a blood test for several patients with diabetes. Data have been standardized. This is a simple example in which we can immediately recognize the presence of two subpopulations located along the main diagonal. However, we do not observe any label for such points. The goal of clustering is indeed to be able to recognize that there are two distinct clouds of points. K-means is a non-parametric machine learning algorithm whose inputs are the number k of desired clusters and an initial partition of the data. The output is a partition of the data in k cluster, which is achieved by iteratively performing the following two steps. In the first step, given a partition of the data, we compute the centroids. Such centroids are simply defined as the center of cloud of data. Let us visualize this with an example. In the image on the left, we have a possible initial partition where points have the same color belong to the same cluster. The initial partition is something decided by the user. We will see in practice that this step is not a difficult choice. Then, this first step is used to compute the centroids, which are represented by crosses in the rightmost figure and are indeed located in the middle of the clouds of points having the corresponding color. In the second step, given the centroids, we update the partition by assigning each point to the closest centroid. Looking at the example, we see that this time the location of the crosses do not change. What changes are the colors of the points. Indeed, all points below the pink line are assigned to the blue cluster and the other ones to the red cluster. What we just described is just a single iteration of the algorithm. Then we iterate the previous two steps until convergence, which means until the partition no longer changes. In general, we do not know how many iterations are needed to reach such final partition. This is why the algorithm is said to be an iterative algorithm. However, we do know from the theory that convergence is guaranteed. It will eventually find a stable final partition. Usually this is a very fast algorithm and only few iterations are needed. Nevertheless, the resulting partition may be different according to which initial partition has been provided. To contrast this problem, we usually resort to a multi-start strategy, which means that we run the k-means for different initial partition and select the best final one.
In the second part of this module, we see together how to fit k-means with R. The dataset we use is a very simple dataset regarding IRC involving 100 patients. Clearly, the goal is to perform a cluster analysis, which means that we try to identify subgroups of patients who responded in a similar way to treatment. To load the data, just use the read.table function with the name of the dataset, which is irc.txt. We must remember that the only input required by k-means algorithm is the number of clusters k we want to partition our data into. The output is a partition of data into such k clusters, which is achieved by iteratively computing the centroids given a partition of the data, and then update such partition given the centroids. The R packages we need to fit a k-means are native in R, which means that we do not need to install any specific package. Nevertheless, to better visualize cluster data, we need a specific library called ggplot2. This is currently the standard to produce nice-looking plots. It also requires the installation of two additional libraries, tidyverse and ggpubr. Have in mind that these are very important and large libraries, and their installation may require long time. Fitting a k-means in R is very simple. The function we use is named k-means. It takes at least two inputs, even though it is convenient to specify an additional one. The first input, named x, is of course the set of data we want to cluster. It is enough to pass the data we loaded from the file. The second mandatory input is named centers. Here we must specify the number of clusters we want. Then, for example, we can set 3 clusters. The third final option is nstart. This option is not mandatory and can be omitted. However, it is better to specify it because we just saw that different k-means starts do not always reach the same final partition. To obtain more stable and robust result, we can specify how many times we want our algorithm to be run. In this case, we are selecting 10 different starts. Moreover, note that the initial partition is automatically computed inside the R function, and we cannot specify it. How do we check if the clustering is reasonable or not? The simplest thing to be done is to plot the data. The code in the slide automatically produces a scatter plot of the data, colored according to the estimated partition, one color for each cluster. These lines of code are not trivial to be understood for beginners, so I will limit myself to show you how to adapt this code to your needs. First, one must set the number of colors to match the number of clusters. We selected 3 clusters, hence we must define 3 colors. This is done writing 3 in the green circle in the slide. The only change you must do is to match the names of the variables of your dataset. This is done modifying the names in the red circles. Going back to our example, the final partition seems reasonable. Each cloud of points has a circular shape and clusters are well separated. This means that subpopulations are indeed heterogeneous. To understand if 3 clusters are suitable for this problem, we can replicate the analysis using different values of k. This can be done using the same code as before. It is enough to change the number of centers passed to the k-means function. In this plot, it is clear that 3 is the best solution. Indeed, k equal 2 is not able to recognize one of the subpopulation. It is unrealistic that all the red dots belong to an homogeneous group. On the other hand, either k equal 4 and k equal 5 do not provide a good clustering because they are splitting homogeneous population, hence providing a final partition with clouds of points that are too similar to one another.